Basic qualities of living organism. That is number one, protection from heat, cold, rain, famines. Number two, growth by physical growth and mental growth. And third, propagation of species. This prevents extinction of species. So basic qualities of living organisms are protection, growth, and propagation of species. Gonads, paired sex organs for production of gametes or sex cells. Gonads, they are the paired sex organ for production of gametes or sex cells. Male sex cells, they are called spermatozoa. Female sex cells are called as ova. Spermatozoa, they are produced in male gonad, that is called testes, and ova, they are produced in female gonad called ovaries. Formation of spermatozoa is called as spermatogenesis, and formation of ova in ovary is called oogenesis. The male gamete, that is sperm, spermatozoa, female gamete, ovum, oocyte, fuse, and this is called as fertilization. Development of human being. Human development, it is a continuous process. The development before birth is called as prenatal development, and the prenatal development is divided into three phases, that is the pre-embryonic period or the pre-implantation period. The second period is the embryonic period, and the third period is the fetal period. So let's learn about the pre-embryonic period or the pre-implantation period. It begins from the fertilization to the end of the second week of intrauterine life. So it begins from the fertilization to the end of the second week of intrauterine life. The events that are occurring in the pre-embryonic period are fertilization, the number two, cleavage, that is mitotic division that leads to the formation of morula. Now, the transport of morula along the fallopian tube towards the uterus. This is that also occurs. Then the blastocyst is formed. Then implantation, that is the attachment of blastocyst to the uterine endometrium. Then the specialization of blastomeres to form embryoblast and supportive nutritive structure called trophoblast. Then these embryoblasts differentiate into bilamellar germ disc, that is two layers. They have the ectoderm and endoderm, and the trophoblasts differentiate into cytotrophoblast and syncytiotrophoblasts. So let's revise again. What are the events that occur in the pre-embryonic period or the pre-implantation period? The fertilization, the period from the fertilization to the end of the second week of intrauterine life. This is called the pre-embryonic period. The events that occur in the pre-embryonic period are fertilization, cleavage, that is mitotic division to form morula, the transport of morula along the fallopian tube towards the uterus, the blastocyst, the formation of blastocyst, implantation, that is the attachment of blastocyst to the uterine endometrium, then the specialization of blastomeres to form embryoblast and supportive nutritive structure called as trophoblast. Embryoblast differentiates to form bilaminar germ disc, that is two layers, and they have ectoderm and endoderm, and the trophoblast differentiates to form cytotrophoblast and syncytiotrophoblasts. Now, let's learn about the embryonic period. The embryonic period lasts from the third week of intrauterine life to the 8th week of intrauterine life. So, embryonic period lasts from the third week of intrauterine life to the 8th week of intrauterine life. Now, the events that occur in the embryonic period are trilaminar germ disc differentiation, that is the formation of three-layer germ disc with mesoderm between the ectoderm and endoderm. The events the trilaminar germ disc differentiation, that is the formation of three-layer germ disc with the mesoderm between the ectoderm and endoderm. Early organogenesis, that is the lungs, heart, liver. The early formation of these organs occurs, that is lungs, heart, and liver. The formation of extra embryonic supportive structures or organs called as placenta, umbilical cord, amnion, allantois, etc. So, what are the three events that occur in the embryonic period? The embryonic period lasts from the third week of intrauterine life to the 8th week of intrauterine life. The events that occur in the embryonic period are the trilaminar germ disc differentiation, that is the formation of three-layered germ disc with mesoderm between the ectoderm and endoderm. Early organogenesis, that is of lungs, heart, liver. Formation of extra embryonic supportive organs, that is placenta, umbilical cord, amnion, and allantois. Fetal period. It occurs from the 9th week of intrauterine life to the 9th month. The fetal period occurs from the 9th week of intrauterine life to the 9th month. The events that occur in the fetal period are the growth of fetus in all dimensions, specialization of various body structures. The events that occur in the fetal period are the events that the growth of fetus in all dimensions and specialization of various body structures. The development after birth is called as the postnatal development. That is from the birth to adulthood. The development after the birth is called as postnatal development. The events that occur in the postnatal development are neonatal, that is the birth, from the birth to the 28 days, that is 4 weeks. That is called as neonatal, and the study, that is the branch of medicine that takes care of a neonate, is called as neonatology. So the branch of medicine that takes care of neonate is called as neonatology. And infancy, 1 month to 1 year is called as infancy. 1 month to 1 year is called as infancy. Childhood is the second year till the 12th year. The pediatrics, they are the branch that deals with infant and children. Childhood is the period from the second year to the 12th year. That pediatrics, that is the branch that deals with infant and children. Puberty, from 12 years to 16 years, puberty. Adolescence, 17 years to 20 years. Adolescence, 17 years to 20 years. Adulthood, 21 years to 40 years. Adulthood, 21 years to 40 years. Middle age, 40 years to 60 years is middle age. 40 years to 60 years is middle age. Old age, more than 60 years till to the death, that is old age. Advances in embryology. What are the advancements in embryology? Prenatal diagnosis, that is the detection of congenital abnormalities in an unborn child. The techniques that are used are amniocentesis, ultrasonography, fetoscopy, fetal blood sampling, maternal serum screening, etc. So the prenatal diagnosis, that is the detection of congenital abnormalities in unborn child, techniques used are amniocentesis, ultrasonography, fetoscopy, fetal blood sampling, maternal serum screening. Next, in vitro fertilization, IVF of the human ova and their transfer in the uterus. The in vitro fertilization, that is the IVF of human ova and their transfer in the uterus. Louis Joy Brown was the first test tube baby. Louis Joy Brown was the first test tube baby. Gene therapy, that is the replacement of a deficient gene product or a correction of an abnormal gene. Gene therapy, replacement of the deficient gene product or the correction of abnormal gene. Cloning, Dolly sheep was the first mammal clone using somatic cell transfer. So Dolly was sheep, first mammal cloned using somatic cell transfer. Cloning, that is the Dolly sheep, first mammal cloned using somatic cell transfer. Stem cell therapy. Stem cell therapy, that is cells have the ability to renew themselves and differentiate into range of special cells. That is, they have the ability to renew themselves and differentiate into the range of different special cells. There are two types, that is ESC, that is embryonic stem cell, and adult stem cell. Embryonic stem cell is isolated from the inner cell mass. They are the pluripotent cells. The embryonic stem cells, they are isolated from the inner cell mass. They are pluripotent The adult stem cells, they are the bone marrow. They are multipotent. They are not pluripotent. The ESC, that is the embryonic stem cell, they are isolated from the inner cell mass and they are pluripotent. At the adult cell mass, adult stem cell, bone marrow, they are multipotent, not pluripotent. Let's learn about some embryological terms. They are oocyte. Oocyte, that is female germ cell, sex cell that produced by ovaries. Mature oocyte are called as secondary oocyte. Oocyte, that is female germ cell or the sex cell produced by the ovaries. They are the mature oocyte called as secondary oocyte. Sperm, male germ cell produced by testes is called as sperm. Male germ cell produced by testes is called as sperm. Zygote, cell formed by the union of sperm and ovum. Cell formed by the union of sperm and ovum is called zygote. Cell formed by the union of sperm and ovum is called zygote. Conceptus, that is the embryo with extra embryonic membrane is called as conceptus. Conceptus is the embryo with the extra embryonic membrane. Cleavage, that is series of mitotic division of zygote to form blastomeres. That is cleavage, the series of mitotic division of zygote to form blastomeres. What is morula? Mulberry shaped, and it is a solid ball of 16 to 32 blastomeres. Morula is the mulberry shaped solid ball of 16 to 32 blastomeres. What is blastocysts? They form from late morula. The blastocyst forms from the late morula. They are of the two parts. There are two parts, that is outer layer of small slightly flattened cell called trophoblast and the inner cell mass called the embryoblast. The cavity of blastocyst is called as blastocele. The blastocysts, that is, they form from late morula. There are two parts, outer layer of small slightly flattened cells called as trophoblast and the inner cell mass called the embryoblast. The cavity of blastocyst is called as blastocele. Then implantation. Attachment and embedding of blastocysts into the uterine endometrium is called implantation. Implantation, it is the attachment and embedding of blastocysts into the uterine endometrium. Gastrulation. Formulation of the three germ layers, that is ectoderm, endoderm and mesoderm, is called gastrulation. Gastrulation is the formation of the three germ layers, that is ectoderm, endoderm and mesoderm. Neurulation, that is the process by which neural plate forms from the neural tube. The process by which the neural plate forms from the neural tube is called as neurulation. Neurulation, that is a process by which neural plate forms from the neural tube. Primordium, that is the beginning or the first indication of an organ or a structure. Primordium, that is the beginning or the first indication of an organ or a structure. Fetus, developing human from the 9th week to the birth till birth is called as fetus. Fetus is the developing human form from the 9th week to the birth till birth. Gestation, the process of development and maturation of the embryo in the uterus from fertilization of ovum until the delivery. The gestation is the process of development and maturation of embryo in uterus from fertilization of ovum until the delivery happens. Let's learn a fact box. Founder of embryology is Aristotle. Founder of modern embryology is Carl Ernst von Baer. The founder of modern embryology is Carl Ernst von Baer. The inventor of first mammal cloning was Ian Wilmut. The inventor of the first mammal cloning was Ian Wilmut. The stem cells were discovered by James Still. Most famous Siamese twins are Chang and Eng Bunker. The most famous Siamese twins are Chang and Eng Bunker. Now, Why do we need to learn embryology? To explain the position and relation of various organs and neurovascular structures in adult anatomy. To explain the various positions and relations of various organs and neurovascular structures in an adult anatomy. Help us understand the cause of development of various congenital anomalies. That is, it helps us understand the cause of development of various congenital anomalies, and helps understand the cause of infertility and its management, and also family planning. It helps us understand the cause of infertility and its management, and also family planning. Helps us understand the development of various embryonic tumor. It helps us understand the development of various embryonic tumor. It provides the basis for medical termination of pregnancy. It provides the basis for the medical termination of pregnancy. And it also provides the insight for the use of molecular biology for genetic regulation of human development. That is, it is used in the molecular biology for genetic regulation of the human development. Now, the diseases that is likely to be benefited by the stem cells are Parkinson disease, Alzheimer's disease, spinal cord injuries, and sickle cell anemia. The disease likely to be benefited by the stem cells are Parkinson disease, Alzheimer's disease, and spinal cord injuries, and sickle cell anemia. Now, let's learn about this diagram. There is menses, ovulation, fertilization, implantation. And from the 2 weeks or the 14, from the menses, the 2 weeks or the 14 days ovulation occurs, and at the same day fertilization occurs. So from that 14 days, the ovulation age or the post-conceptional age starts. So that is 38 weeks. Now from the menses till the implantation, the gestational age or the menstrual age occurs, that is the 40 weeks or 280 days. Now from the menses, the implantation occurs at after 3 weeks, that is the 21 days. So gestational age is nothing but the ovulation age plus 2 weeks. The gestational age is nothing but ovulation age plus 2 weeks. Implantation occurs at the 7th day post fertilization, like post fertilization or after ovulation. After 7th day, that is after 2 weeks are already passed after menses, ovulation occurs, and after another 1 week the implantation occurs. After another 1 week implantation occurs. Now we classify the embryology. The classification of embryology, that is general embryology, systemic embryology, descriptive embryology, experimental embryology, chemical embryology, comparative embryology, and teratology. Now what is general embryology? Development of individual during the first 8 weeks after fertilization period is called as general embryology. The general embryology is the development of individual during the first 8 weeks after the fertilization is called as general embryology. What is systemic embryology? The functional maturation of various organs and the systems of the body during embryonic period is called as systemic embryology. That is basically the study of the maturation of various organs and the system of the body during and the embryonic period is called systemic embryology. Descriptive embryology, the study of different organs at the different maturation or the different various stages of development or different maturation levels is called descriptive embryology. The study of the structure of different organs at various stages of development is called descriptive embryology. Experimental embryology, it is the results obtained from the experiments of living embryo and fetus of lower animal. So it is the result obtained from the experiments of living or embryo and the fetus of lower animal. Chemical embryology, that is biochemical aspect of the prenatal development. The biochemical aspect of the prenatal development is the chemical embryology. Chemical embryology is nothing but the biochemical aspect of the prenatal development. Now what is comparative embryology? The comparative embryology is nothing but the study of embryos in various species of animals. So the study of embryos in various species of animals is called as comparative embryology. Comparative embryology, study of embryos in various species of animals. Now what is teratology? Teratology, it is concerned with the congenital anomalies or the birth defects. That is, teratology, it is concerned with the congenital anomalies or the birth defects. Okay. Thank you.